Our next speaker is Hosseinali Asgharian from USC.
Thank you. Today I'm going to talk about an ongoing project we have to use population genetic theory to estimate certain cellular aspects of body regeneration in Dugesia platforms. So Dugesia is a genus that is a part of a group of free-living platforms collectively known as Planaria. They have been known for more than 2 centuries for their extraordinary capacity for regeneration. But apart from regeneration in response to wounds, the asexual reproduction cycle for some of these worms includes just growing in size, splitting in half, and then each half form can reconstruct the whole body. But why did we choose this model for population genetic study? 2 reasons. First, there are some questions, important questions about regeneration that molecular biology methods have so far failed to answer satisfactorily. For example, it is not known how many stem cells exist in the body, what % of them are active during each regeneration cycle. Is there just one type of general stem cell or specialized subclones that are each adapted better to produce certain types of tissues and organs? On the picture to the right, you can see one planarian worm stained for 2 markers for stem cells, one in red, the other in green. The fact that we see plenty of red and green spots in addition to yellow spots shows that the 2 markers do not completely agree. In other words, the stem cells are heterogeneous populations in terms of molecular markers. They also overlap with early committed cells on the pathway to differentiation. These worms are also interesting from an evolutionary perspective because they are clonal reproducers. They do not undergo that single-cell bottleneck that is zygote, which means there is high level of genetic variation among somatic cells in one worm's body. And that means we can expect a high level of competition among somatic cells. Add to that the fact that these guys do not have recombinations, so mutator alleles will remain with the mutations they cause, and also somatic mutations can be transmitted to next generation. Therefore, the question is, what prevents competition among somatic cells, disrupt the structure of the worm's body, and create cancer all the time, or what is the optimal mutation rate, somatic mutation rate, for these worms compared to sexual reproducers? So the idea for this project was to basically model each cell as an asexually reproducing individual and the body of the worm as a population of asexually reproducing cells. Because we know that there is variation among somatic cells, we can look at the fluctuation in allele frequencies of these somatic variants across generations, for example, to estimate effective population size for these cell populations. And because we know that in planaria, somatic differentiated cells never divide, effective population size in this case will actually mean the number of active stem cells in the worm's body. By the way, this is not the picture of the worms we work with, but this is a more beautiful species, so it made it to the presentation. So the theory to estimate effective population size from observed changes in allele frequency was developed in the 80s. The idea is to calculate a F statistic based on the difference in observed allele frequencies in samples taken from the population at generations I and J, for example, PI and PJ, and then through the formula at the bottom, we can connect it with effective population size and the number of generations that has passed. It has Si and Sj sample size at generation I and J, which gives the component of variance due to sampling error, and the term in the middle has Ne and T, which gives the component of variance due to genetic drift. But how does this correspond to our model system? We have kept these planarians in the lab for several generations. After each natural fission, the tail piece is frozen for future experiments. The head piece is allowed to grow and create the whole worm and further the worm lineage. We have taken tail pieces for sequencing every other generation 8 times, which means we have sequences from generation 1, 3, 5 through 15. In this system, basically because we're using the head piece to further the lineage, effective population size would mean the number of active stem cells in the head piece. Sample size at generation I and J would mean sequencing coverage for each locus at generations I and J, because we know that each sequencing read will come from a unique chromosome in the worm's body. So because we have sampled 8 generations, we have basically 8 genomic data sets. We have used a reference-free method for discovery of SNPs, disco SNP, and based on that, we have found 360,000 SNPs in this data, this sequencing coverage. across polymorphic positions per sample was between 22 to 25, which means we have 160x coverage or more combined in all samples. So we have good power to detect the SNPs, basically. Excuse me. The first question is a kind of proof of concept question. Does this data set really contain any information on drift that we can use to estimate population size? The method to answer that question is a simple heterogeneity chi-square test. For each locus, we can construct a 2x8 contingency table and count the number of the 2 alleles that we have at that locus across generations. If the result of this chi-square test is significant, it means that we have more fluctuation in allele frequency that can be accounted for merely by sampling error, which means we are observing genetic drift. So we subsampled 100 loci randomly from the data set, and the figures below show the histogram of p-values and FDRs. 54 out of the 100 have p-value below 5%. 45 out of the 100 have FDR below 5%. This basically shows that enough number of our loci contain information on genetic drift, and we can use this data set to estimate population size. And these are our actual estimates. The plot on the top shows estimates based on different pairs of generations. We have sampled 8 generations. We can do the estimation based on any pair. We have done the estimation based on all 28 possible pairs of generations. And as it can be seen, it varies greatly. It basically varies from 10 to 900. We believe the main reason for this high level of variation is the place where fission happens. Because we are using the headpiece, if the fission happens very close to the anterior end of the worm, it means a smaller number of stem cells. And generally, the headpiece is known to have fewer stem cells than, the posterior end. The area anterior to the eyes is known to be completely devoid of stem cells. So it matters where the fission happens in the worm. Part of it also can really be biological stochasticity in terms of the number of stem cells that are activated during each regeneration cycle. And then we decided to check that by tracking the amount of nucleotide diversity we have in these different 8 generations. Because it's a reference-free method, we only find information on SNPs. We cannot directly calculate per position pi for the whole genome. What we can do is to calculate the average of pi across polymorphic positions. And that is what we call pi star, and that is on the y-axis on the graph at the bottom. And it is interesting to see that the biggest drop in the level of diversity between generations 6 and 7 also coincides with the smallest estimate of population size, which is 10. Unfortunately, this association does not hold for all of the pairs of generations, but at least for the biggest drop and the smallest effective population size, it can be seen. I shouldn't forget to mention that these estimations were not based on 100 loci. This was based on a data set of 10,000 loci that we picked initially and then filtered for removing extreme allele frequencies that are known to bias this estimate, and also for coverage. So what else can we calculate from this data set? We have this formula pi equals 2n mu, basically because each sequencing read is coming from a haploid chromosome. This is not 4n mu. This is 2n mu in this case. I had mentioned that we cannot directly calculate pi. However, what we can do is to multiply pi star, pi across polymorphic positions, by the proportion of polymorphic positions in the genome, if we can somehow estimate that. An order of magnitude estimate we can make is to divide the number of discovered SNPs by the genome size. So we have discovered 360,000 SNPs. We know from running this method on simulated data sets that the recall success is almost always above 80%, and the precision success is above 90%. So basically, we know that there are not many false positives and false negatives using this method. Genome size was taken from a congeneric species. Our species is Dugesia gonocephala, but this genome size is from Dugesia japonica. Also, the genome size for a better studied planarian Escherichia mediterranea is 0.8 gigabases, so we can be pretty confident that this is the correct range, the correct order of magnitude estimate for genome size. Based on that, pi will be in the order of 10 to the minus 4. We need a single value for effective population size. We took the harmonic mean of NE for the different pairs of generations, consecutive generations. and based on that, NE turned out to be 19, and then we can calculate somatic mutation rate, and that is in the order of 10 to the minus 6. Now, this is a much higher rate than we normally observe in other organisms, which are often in the order of 10 to the minus 8 or minus 9. If this is a true order of magnitude estimate, there can be biological explanation for it. There is theoretical work, mostly from Sarah Otto's papers, that shows that if there is a strong selection among somatic clones in an organism's body, then we can expect a higher optimal mutation rate, because mutator alleles can benefit from the beneficial mutations they cause, but the deleterious mutations can be selected out and can be eliminated before they get the chance to be transmitted to the next generation. So it is possible to have higher somatic mutation rates if there is somatic selection. Finally, we tried to calculate Tajima's D from this data set. Because these guys do not have meiotic recombination, we can imagine the whole genome to be a single giant locus. So if we do that and calculate Tajima's D for that big locus, it turns out to be 2.4. Now, when we see such a positive big number, we go back to check the site frequency spectrum, of course, and the plots below show site frequency spectrum for 3 out of the 8 generations we have sequenced. And as you can see, it is completely opposite to what you expect from neutral theory. Instead of having mostly rare variance, we can see a peak around frequency 0.5. Okay, so we are pretty confident. Again, this is not a computational artifact. This method, DiscoSNP, is known not to be biased against rare variance. So if there is a biological explanation, what can it be? Normally, positive Tajima's D is interpreted as evidence for balancing selection. However, this is a whole genome estimate. So imagining balancing selection due to selective force on all loci or majority of loci one by one is a little unlikely. There is more likely to be an explanation based on body structure here. A peak around 0.5 is consistent with 2 different genetic clones. It can be 2 different cellular clones. In an experiment with single-cell transcriptomics in planarians, it was shown that they actually contain 2 separate clones of stem cells, 2 different types of stem cells. But in that experiment, they concluded that they shared recent ancestry. So that may or may not be explaining this observation. Another interesting possibility is also that because they do not have recombination, we are looking at 2 genetically diverged sets of homologous chromosomes. With the data we currently have, we cannot distinguish between these hypotheses, and I'm going to say what plans we have to be able to get the data that can answer these questions. So the next step in this project, firstly, is to do the same estimations using simulation-based methods, basically ABC, instead of doing it analytically. One of the reasons for that is our different estimates show that they are not necessarily completely consistent. When we see a positive value for Tajima's D, it shows that the genome is not acting in a neutral way. Therefore, the equations we used for estimation of mutation rate may not be completely valid. Also, we have assumed in our method for estimation of effective population size that there is random selection of individuals to be members of the effective population size, the members that are reproducing, which means activation of stem cells in a random manner. On the figure to the top, it would mean the pattern to the left, but the stem cells might be activated in a more structured, uniform way in the body of the worm, or they may be activated in a way that is inversely proportional to their distance from the fission site. And these different scenarios are much easier to put in a simulation-based estimation scheme rather than tracking analytically. Also, there are different patterns of sequence and number of symmetric and asymmetric division of stem cells, which eventually produce the same number of stem cell and somatic cells, but they will have different consequences on observed temporal variance in allele frequencies. Again, this way will be easier to estimate through simulations. Also, experimentally, we plan to sequence smaller fragments of worms' bodies rather than whole half bodies. And doing that with knowledge of their position in the body, we can do population structure, basically, on these worms and answer some of the questions I mentioned on previous slide. This was an attempt to use population genetic theory basically to connect cell-level genetic data to some organismal features. And hopefully, when single-cell sequencing data become available population genetic theory will be one of the favorite tools for analyzing that kind of data, and then hopefully that would mean better funding and more positions for population geneticists. Finally, I would like to thank my advisors, Sergey and Paul, and collaborators on this project, Joe and Hamdi, USC and NIH for funds, Worms for being awesome model organisms, and GSA for giving us the opportunity to present our research. Thank you.
Thank you. Okay, our next speaker is Alexander Platt from Temple University.